So now we've got the structure of the membrane down. Now we've got to talk about the function, how it works. So all of that chemistry that we just talked about comes into play with transporting molecules across the membrane using diffusion, osmosis, or active transport. Selective permeability is a critical role played by the plasma membrane. It allows certain things into the cell, but not others. It's sort of like your relationships, I assume. You want some things to come in, you want some people in your life, you don't want other people in your life. Especially true now on like social media. You're like, block, block, block. If you haven't done that yet, it's totally good. You don't have to see whatever these other people say. It's great. So you want to allow some things in, but not others. Because if you allow too much in, you're going to get toxified. You're going to get a lot of bad material in. If you're going to let too much out, you're going to end up releasing your nutrients. And you don't want to do that. So the cell membrane prevents things from getting, prevents nutrients and substances from getting out that you don't want out, and prevents them from getting in that you don't want in. There's a few ways across with diffusion, facilitated diffusion, and active transport. The key to this whole thing is that hydrophobic center of the phospholipid bilayer. We've got a hydrophilic head, a hydrophobic layer in the center, and then a hydrophilic layer. It's sort of like Tootsie Pops. If you look at a Tootsie Pop, you've got this candy outside, hard candy outside, and this chewy candy in the inside. They have completely different properties. Whereas it's easy to bite down on the center part, it's harder to bite down on the exterior part. So I've noticed, and maybe I'm wrong, a lot of people may like the outside part, or they like the inside part, but the two may not go well together. Oh, you know what else is like? What are those things? So blow pops. You have a blow pop and you're like, I would like some gum. Well, you can't have it. It's trapped inside of that candy. Well, I just want the candy. Well, I don't want gum. So you've got two totally different layers going on, each with distinct properties. And molecules interact with both distinctly. Some molecules are highly permeable. They go right through. They go right through the membrane as if it didn't exist. Gases and small uncharged particles can fit between the different phospholipids. They're small enough to slide through. It's almost like, I was at an amusement park with my son, and he's small, and I'm bigger than him. Surprise. So he was able to negotiate his way between the crowd and get far, far away from me. I couldn't. I was stopped by the larger particles, the larger people. Small things can get through places that other things can't. In this case, gases do. I'm not sure how much you know about the circulatory system, so I'm going to give you a super brief, here's everything I know about anatomy, because I really don't know much. Anatomy is not my thing. You breathe in oxygen, right? It comes from out here in your lungs. And there's all sorts of ways it gets there. It could go through your nose, but if your nose is stuffed, it goes through your mouth. You can get it there all sorts of ways. Oxygen goes down to your lungs. In your lungs, you've got blood vessels down there that are really close to the surface, so that oxygen can go from high concentration in the lungs into the blood cells. So it sort of like sidesteps in. And then the blood cells carry that oxygen. Now, the only reason they were able to move is because there was a low concentration of oxygen in the cells, high concentration in the lungs, and it moved over. And then as those blood cells start moving through your body, because you got this part and your heart pumps things, they're sort of leaving that oxygen behind, almost like a, if you've ever cooked pasta. You've cooked pasta? Yeah, good. All right, you take pasta. It's in a pot. In that pot is pasta and water. You take it over to a strainer. Where's the strainer located? That's the best answer. Not always the one I go with for some stupid reason, but it works. You take it to the sink. You pour the bowl into the strainer and you lift it up. What's coming out of that strainer? Water. Now you got to get the pasta from the strainer to the bowl that you want to put the pasta in. Where's that pasta bowl? Hopefully, right next to the sink, right? Sometimes, if you don't plan well, it's like in the cupboard or it's all the way across the room. So you take this thing of pasta with the strainer that's leaking all of this water. You go across the room and you dump it into the bowl. You have now left behind a trail of water. Does that make sense? Not if you're trying to catch the boiling water. That water dripping out is exactly what would happen from the strainer, is exactly what would happen to the red blood cells. Oxygen's constantly streaming out of them, going to the cells around it, until they finally get to their destination where they slow down a bit. And we'll get into that in like 102. We'll talk about capillaries and we'll talk about webbing and how everything slows down. But once it slows down, the oxygen finishes diffusing. It goes to the area of low concentration in the other cells. And it picks up while it's down there carbon dioxide, which then goes back to the lungs. You get these blood cells back up to the lungs, and they push this carbon dioxide from inside of them, where you have a lot of carbon dioxide, to the lungs, where there's a little bit. And it proceeds right across that membrane as if there was no membrane there at all. Super highly permeable. Gases and small uncharged molecules. It goes right across. It's like the membrane doesn't exist. Water's pretty darn permeable. There's a bunch of proteins in the membrane called aquaporins. And what aquaporins do is they are just big enough to allow water molecules through. Not to allow bigger molecules through, just water. They're selective in that way. And when they've got a hydrophilic region that sucks them in, almost like a little water vacuum. They can pull billions of water molecules across a second. So it's as if this membrane was totally permeable to water. Water's just constantly flowing in, and it's constantly flowing out. Only water. And then you get more molecules. They get bigger. They get less permeable. Polar molecules, sugars, they're easily dissolved. They can get in sometimes. They can fit through channel proteins, but it's getting harder and harder to get in for them. Why? Because they're a little bit too large to fit, or they may, when they hit the phospholipid bilayer, they're interacting with that hydrophobic layer and it's excluding them, it's pushing them out. Oh, definitely. Not all molecules are the same size, without a doubt. And these molecules, amino acids, proteins, nucleic acids, polysaccharides, most of our macromolecules, they're too big. They won't fit. So they can't make it through that membrane at all. So you have gases, small uncharged molecules, and water that can very easily cross that barrier. Just about everything else needs help. They could make it through, but they need help, and we're going to talk about what helps them through. Before we get there, we're going to talk about small molecules moving along what's called their concentration gradient. A gradient is where you have a high concentration in one area and a low concentration in another. Solutes will always move from a high concentration to a low concentration automatically. No energy required, always happening, until they're evenly spread out. That process is called diffusion. If you guys ever, you've had to have, if you've made Kool-Aid or you've made some sort of instant iced tea mix, you put the iced tea in the container or the Kool-Aid in the container, it sinks to the bottom. That is a high concentration at the bottom. You wait for a day or 2 days because you don't want to stir it on your own because that requires energy. Over time, those molecules evenly distribute themselves throughout the entire solution. Thing is, if you were to put a membrane right in the middle, that's where it mucks things up. Before I go on, you guys do remember what a solute is, right? Talk about, sorry. You're like, yes, move on. A solute is a particle that's been mixed with a solvent to create a solution. If I was to take salt and then put it into water, I get a saltwater solution. The solute would be the salt. So we're really talking about solute concentrations. Ooh, and what unit of measurement did we have to measure solute concentration? Moles. That's why we learn moles. But the thing is, it's not just the amount of solute that's present, it could be the charge. You'll notice here we've got an equal number of solute on either side. But on one side, we have more positive charges, on one side we have more negative charges. When you have difference in charges on either side of the membrane, you've created what's called an electrochemical gradient. So you have charges on this side that are positive, charges on that side that are negative. They want to even out. So what you end up having with an electrochemical gradient is a need to move molecules from one point to another to even the whole thing out. These positives need to come over here. Those negatives need to move over there. That creates a flow. And in biological systems, we can use that flow to create energy. This is ultimately the basis of cellular respiration. Right. If one side of the barrier has a lot of positive charge molecules and one side has a lot of negative charge molecules, they want to shift sides so it's equal. And you can use the energy of that movement to power your biological reactions. These gradients can also move water across a barrier because it's really hard to move solutes across a barrier. We just went through everything that can't get across the membrane. Water can, though. So in order to balance out solute concentrations, to reach that equilibrium, water can move from one side to the other. So over here we have hypertonic, sorry, words you don't know yet. We have lots of solute. Yes?
Sorry,
no, please.
So when there's a net positive and negative charge on one side but not the other is when that occurs?
Yes, that's exactly when it occurs. When there's a net positive charge on one side and not the other for an electrochemical gradient. So here we can use gradients to move water. Here's our semi-permeable membrane. It'll let some things through but not others. It'll let water through, for instance. On this side, you'll notice there's some molecules that are just too big. They still want to have roughly the same molarity. So what will happen is the water will rush from the side with less solutes to the side with more solutes. Membrane. We can describe these solutions in different ways. If we have two solutions that have equal amounts of molarity, equal amounts of solutes per unit volume, they would be termed isotonic. Iso means same. Isotonic. Isosceles triangles all have the same angles. Isotope, isomer. So iso, we have the same solute concentrations. Hypotonic, hypo means small, a hypodermic needle. Hypoglycemic. You have less solute in solution than you have in the cell. Right, so hypo is small, less solute. If you're less solute in solution than in the cell is hypotonic. Smaller amount of solute. And then we have hypertonic. What does hyper mean? Lots. If you're hyperactive, lots of energy. In this case, there's lots of solute outside the cell, not so much inside the cell. So hypertonic, lots of solute in solution, not so much solute comparatively in the cell. So it could be isotonic. Two solutions could be isotonic to each other. One could be hypotonic to the other, which is hypertonic. Water will always move from hypotonic to hypertonic. That's a statement you're just going to have to remember, and it will solve a lot of your problems. Water moves from hypotonic to hypertonic. Because we can't really move solutes that well. There's a membrane in the way. Here we have a hypertonic solution. Here we have a hypotonic solution. Water is going to move from this hypotonic solution into the hypertonic solution. And it will actually shift these areas. There's a membrane separating them. Always hypo to hyper. Water moves hypo to hyper. That water part's important. Yes, Olivia?
You said water moves from hypertonic to hypo.
Water will always move from hypotonic to hypertonic. Water moves from an area of low solute concentration to high solute concentration. That movement of water is called osmosis. It balances out solute concentration. Osmosis Jones. Yeah, it had nothing to do with osmosis. I watched it just to find out about osmosis. Didn't work. Osmosis is a type of transport that's termed passive because it requires absolutely no energy. There's active transport that requires energy. This is called passive. So here's an example. Water is moving from the solution into the cell. So how would you classify the solution? Hypertonic, hypotonic, or isotonic? Hypo, because water moves from hypo to hyper. Here, what would you classify the solution? Water's moving out of the cell into the solution. Hypertonic. Water always moves from hypo to hyper. And then this one, water's moving evenly in both directions, so it's isotonic. Would I give you one that had, I don't know, 2 molar on the outside, 1 molar on the inside? Which way would the water go, out or in? 2 molar on the outside, 1 molar on the inside, which means there's a lot of solute on the outside. So the water will flow, the water's going to flow toward the hypertonic solution, the one with the more solute. That's stupid. Sorry. In animal cells do not have a cell wall. So there are 2 consequences of putting cells into solutions that are not isotonic. Animal cells do not do well outside of isotonic solutions. You might have osmotic lysing, or you might have crenation or crenellation. Both are the same. With osmotic lysing, water will move from a hypotonic solution into a hypertonic solution. If you were to take pure water and surround cells with it, the water would flood into the cells, causing them to eventually pop. Sort of like water balloons. When you guys, I assume you've thrown water balloons at people, with or without their knowledge. That's cool. Do you fill them up at the sink, or do you fill them up at the hose? The hose? Okay. If you got a little funnel, there you go. So you take these water balloons, you fill them up at the hose. First type of water balloon you fill up, you only fill it up part way, you tie it off, you throw it at somebody, it bounces off them. And then you've lost the element of surprise. Second type of water balloon is the perfect water balloon. It's filled up just right. You throw it at them, on impact it explodes. The third type of water balloon, though, is the one we've all experienced, which is osmotic lysing. You fill it up, and you think this is going to be the best one ever. It gets bigger and bigger and bigger until what happens? It pops. Because the structure of the outside cannot contain the water on the inside.
So did you say it goes from hypo to hyper?
Water always moves from hypo to hyper. So if you have a hypotonic solution outside and a hypertonic solution on the inside, water floods in.
Wait, could you repeat? Because you were explaining to her a question. Could you repeat?
Yes. So water is going to move from a hypotonic solution to a hypertonic solution. Outside of this red blood cell is a hypotonic solution. Water will flood into the red blood cell. It will fill up like a balloon, and it will eventually pop.
But you just said that hypertonic was on the
hyper is on the inside.
Okay, I thought
Hypo is on the. Sorry, they sound very similar. I'm sorry about that. I'll try to enunciate better.
Yeah, you're fine.
For crenation, in this case, you've got a hypertonic solution on the outside and a hypotonic solution on the inside. So which way does the water flow? Out. In this case, the cell shrivels up. Yes?
So does water not diffuse evenly? Doesn't seem like it does.
Water diffuses evenly. The problem is these membranes are going to stop these solutes from getting out. So water is going to diffuse in order to make it all even. So
Well, it's about the solutes then.
this is about which way the water's moving because of the solutes. Yes? Okay, I'll wait. Okay.
How do you know which is hypertonic and which is hyper?
You will know based on the molarity. The higher molarity is going to have more solutes, and which means it's going to be hypertonic. The one with the highest molarity.
And you said for the second one that the hypertonic solution's on the outside,
Right. So hypertonic's on the outside, so water leaves the cells,
and that's what makes it dry up.
and that's what shrivels it up.
You
Yes.
said highest molarity is hyper.
Hyper.
What's
Um,
that gonna look like? Because I'm confused.
Uh,
Like, how are we gonna know, like, what the higher?
if I give you, I'll either give you numbers that say this is 2 molar solution, this is a 1 molar solution. Which way is the water moving?
Okay, so that would be 2 to 1.
That would be actually it'd go from 1 to 2. Which way is the water moving?
So you said moving.
So 1 molar solution would be hypotonic. Yeah. Always remember hypo to hyper for water. Question? Right. Okay, the way you can think about this, slugs. If you were to take a slug and drop it in a bucket of water, water will flood into the slug and the slug will blow up like a little sphere of the slug. Go and try it if you want. So water floods in. That slug is acting like a cell. What happens if you put salt on a cell? You create a hypertonic solution around the slug. It dries out, it shrivels up. I assume you've done that. I mean, maybe you're not a horrible person. That's cool. Whatever. I do it to protect my jack-o'-lanterns. Take a ring of salt, put it around my jack-o'-lantern. It stops all the slugs from eating it. What, a ring of salt around it? Oh, good point. Always thinking. Yep. Now, plant cells have a different reaction because plant cells have a cell wall. And that cell wall is made of cellulose. It's made of interlinked carbohydrates that make it really, really rigid. So if you were to take a plant cell and put it in a hypotonic solution, pure water, the plant cells absorb that water and become what's called turgid. Very rigid. They have a lot of pressure. That's what, when a plant is wilty, what do? You give it water. The water goes into the stem, it makes everything more rigid and gives them much more strength. The flip side is if you were to add a hypertonic solution to a plant. Salt water. You guys did that in lab, I believe. Add salt water to a plant, the plasma membrane rips away from the cell wall and puts all the organelles in one corner. It's lost water. It can't survive. It wilts and dies. That's why if you were to, I don't know, try watering plants with Gatorade. I had a student decide to do that one year because, you know, Gatorade's got electrolytes in it. All the plants died across the board because there's too many solutes. The Romans and the Carthaginians, how many of you guys have taken world history or I guess Western Civ? All right, do you remember it was the Pyrrhic Wars, wasn't it? Where the Roman Carthage is in Africa, Rome is in Italy, they fought each other. The Romans beat the Carthaginians back down to Carthage and they're like, all right, you stay there, we're going to go back to Italy and chill and take over the rest of the world. And they're like, okay, that's cool. A few decades later, the Carthaginians were like, hey, let's go mess with Rome. So then they went across the Mediterranean, attacked Rome. Romans beat them back to Carthage, and Romans got there and were like, okay, we said stop. Just stop. Okay, no, we're not going to do anything. Carthaginians then said, okay, we got a new idea. We're going to go and attack Rome, but this time we're going to go up. We're going to take our elephants with us, because that's a great idea. Take our elephants up through Spain, cross through France, across the Alps, down the Pyrenees Mountains, and hit Rome from behind. And they did that, and a lot of their elephants died. But they got to Rome, and then the Romans beat them all the way back down to Carthage. And this time they said, okay, you're done. We're going to salt your fields. So what they did was they took salt crystals, laid them out in the fields, and that salt created a hypertonic solution. So now the plants wouldn't grow. And the plants wouldn't just grow for a year, 2 years, decades and decades. There was famine. The Carthaginians did everything they could to survive, and it was really hard for them. They couldn't make war on Rome anymore. Similarly, my crazy aunt, she decided, she's one of those people who's really into conspiracy theories, and decided that she was going to make her own herbicide. So she made it out of salt. And she decided to spray all of her garden with this salt herbicide to kill the weeds. And she was shocked, utterly shocked, that every single vegetable in her garden also died. And she's like, I made a super herbicide. And I was like, no, you just, literally, thousands of years we've known this would happen. But the Illuminati, so you know. If you were to put a plant into a salt solution, they are going to die because the water gets removed from the cells. If a plant loses 40% water weight, you cannot recover it. For us, if you've been dehydrated, people tell you drink more water. If you go to the hospital and you're dehydrated, they don't hook sterile water up to your arm. What would happen if they put sterile water in your arm? You would scream, ah, they put a needle in me. Why would you die? Why would it be bad? What would happen to your cells? You got pure water going next to these blood cells. They would lyse. Every single one of them starts popping like a little balloon. And it's really cool to watch. Unfortunately, we can't do bloodborne. We can't play with human blood in here because of possible disease. But hypothetically, if you were to look at a drop of blood in a drop of water, what you would see, literally watching under the microscope, is pop, 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 little fireworks going off as all of the blood cells lysed. Water rushes in, causing them all to pop.
Are you able to do like blood cell stuff at like VCU? Are you just referring to like here?
I think for Bio 101 students, it's not a good idea to do it with. That would be fine, yeah. It works with animal blood too, yes. What would happen if you put a red blood cell into a hypertonic solution? All the water goes out. You need to make an isotonic solution, which is why they give you, when you're dehydrated, saline. Because what saline is it's has the same level of solute as your blood cells, as your plasma.
Is that why you can taste it when they like flush?
Maybe. I've never tasted in the back of my throat when they did that to me. Animal cells need to protect themselves from this osmotic lysing. The way they can do that is with a contractile vacuole. Vacuoles are important not just for storage, but they're important for maintaining cell volume. What happens is water floods into the cell and it expands. And then after it expands, the cell uses energy to squeeze it shut and push the water out. Contractile vacuole. So that's osmosis. Osmosis is just sort of the natural extension of diffusion. Moving from an area, solutes moving from an area of high concentration to low concentration. So these are two distinct processes, diffusion and osmosis. Osmosis is the movement of water. Diffusion is the movement of solutes. Osmosis, you're moving from, water moves from hypotonic to hypertonic. Diffusion, you're moving from an area of high concentration to an area of low concentration. And it is passive. It happens without any energy. Here, you've got individual molecules of dye. high concentration on the left, low concentration on the right. Over time, they reach equilibrium where they're equally spread apart. So diffusion is the movement of solutes from an area of high concentration to low concentration. Here's an example. Okay, before we go, what color is this? Blue. Okay. Whoever I hear first, that's the color we're defining it as. What color is this? Green. What color is that? Red. Okay, good. And the reason I have to define those is because I once ended up having the class spend 15 minutes arguing over colors. So these blue ones, which way are they going to move? Toward A or B? They're going to move toward B. High concentration here, so they'll expand out over here. How about the green ones? They're not going to really go anywhere. They're already in equilibrium. And the red ones? They're going to move toward A. That's diffusion.